blood service, Professor University of Helsinki, and is really an expert in blood donors, biobanking, and genetics, and actually immunology as well. And so, Jukka, here's the floor.
Okay. So, first of all, thank you, and Emmanuel, for inviting me here. It's always great pleasure to be here in this beautiful place, and also telling what we have been doing or what we are doing in Helsinki with blood donors and so on. So I think I was safe that I was somewhere here. So no, yeah. So I'm coming from Helsinki, Finland, and it's I'm coming back to Finland also. But here is just the main topics of this talk. I'm a little bit telling of the blood service in Finland in general, just to give you a background, and then something I feel that is quite important, that we are not only assuming that the blood donors are willing to give samples to research. We have been doing some social science studies related to that topic, and then something about the biobanking and genomic studies. And how we are using. How we are utilizing. I think. Yeah. Yeah. Okay. So Finland is there in north, quite far away from here, in northeast side end of the Europe, and we have one blood service there. It's the only one that is in practice. We don't have any monopoly, so anybody can found their own blood service if they want, but in practice there's only one. And we are serving the whole population, and it's only 5.5 million people, so it's only part of London or something. So it's not a huge population. But the size of the country is I guess it's about the same as the free size, perhaps, at least the length of the country. I don't know, I didn't check it, but anyway. We are part of the Red Cross. It means that it's the, in a way, it's the owner of the blood service, but we are independent in administration and in budgeting and so on. It also means that we are not receiving any funding directly from the government. So in a way, we are private, or we are society, if you want to say it that way. The Red Cross has their own legislation, in fact. And we are not for profit. It means that we don't share any of the owners of the blood service don't get any shares or any benefits from the function. And of course, it also means that we spend the money we receive, and most of the money is coming, of course, from red cells, platelets, plasma, and we also have quite active lab systems. We are doing all tissue typing for transplantation, for clinical transplantations in Finland. We have stem cell registry, and we are also involved in organ transplantation. So when we are talking about donors in Finland, we are not only talking about blood donors, we also have stem cell donors, we have organ donors, and also biobank donors as well. There's about 500 people working in a blood service in a whole country, most of them in Helsinki area, but then we have the donation sites in about 10 places in Finland. We have our research budget is not large, about 2 million euros. It's for the academic research mainly. Then we have the separate one for the development and so on, R&D. And actually, we are located not in the same place as the basic blood service production site is. We are in a campus. It's in Helsinki very close to the university hospital. And what else? Yes, about half of the funding is coming outside, so we are spending a lot of time to get some funding outside. There's about 10 doctoral level scientists plus minus something, and the same number of PhD students, depending on the time and how you actually regard them. Some of them are just part-time and some of them full-time. And here is also some numbers. I think I can actually try to. It's a little bit smaller. I say most of the income is coming from platelets and red cells, and then there's some laboratory plasma and others. These are the key numbers in a way. We need something like 100,000 blood donors to donate the blood, and they are donating some of them more than once, so it's about 180 donations each year, and about half of the donors have consented to biobanking, and we will come back to biobanking. So that's what it means in Finland, because we have the legislation that is regulating how you create the biobank. And this is just an overview of the research we are doing, of the topics, these two sides. On the right-hand side, there's novel products. It's related how we are utilizing the donated blood. We are trying to get something some new products out of the blood, and also we are involved in cell therapies. We are producing CAR T cells, and we are also doing some research and R&D related to CAR NK cells, so that we use these. We could produce these cells for hospitals, and we are doing it at the moment also. So we really produce some CAR T cells for hospital use. And then the left-hand side is more related to how to be a better blood service in a way, so that we are doing everything in the right way. So there's something related with blood donation and the data we are producing. And basically it's this guy here, Mikko Arvas, who is the PI. He's one of the data scientists we have. And then we have the blood donor biobank. Then there's something related to virus, of course, it's always a topic. And then one topic that most of the blood banks are, what we are doing is that we are taking cells from one individual and we are transplanting or transfusing the cells to another individual. And there's always a question of matching. It can be blood types, but it can be also organ or stem cell transplantation and how you match them. And that's one of the topics we are also having in the science part. So, yeah.
I'm going to turn off your video. Some people are saying online that the sound is breaking up. So we're just, you're probably going to just stop that. And hopefully the bandwidth is a little bit better.
So, yeah.
Yeah.
Yeah, that's always better that way. Okay, so the first question is I think all blood bankers are feeling that yes, blood donors are very willing to be part of the research, and that's perhaps true, but we wanted to a little bit more see if I can get it here. Okay. We wanted to start something like a sociological study. Do we really know how blood donors are regarding if we are asking them to give samples to research or biobanking, is it the same as giving your blood to hospital that is in kind of patient care, something that is really needed? And we hired a student, Veera Raivola, some years ago already, and she actually, her thesis is still a little bit waiting for the finalizing, but anyway, together with some people in Finland who are more in the social science, we started a project that is related how the kind of gift relationship that is the classical thinking of Titmus. He was one of the main persons who he was thinking how the blood donors are actually feeling when they are donating, and it was kind of gift to the society, and they were thinking helping others and actually for help when they needed. It's kind of relationship. How this kind of paradigm was working when you are asking blood donors to give samples for research. Is it the same? Are they feeling that yes, it's the same thing or not? And we have been doing some publications, and they are quite old already, but you can read them if you want. But there's some key points here. What Veera was actually doing in practice is something called qualitative sociology. I was much educated for these topics, and when I was discussing with these people, my background is in biology and genetics, and this was very interesting. And she interviewed these donors and collected what kind of different thinking they have and how they felt, and there was a lot of interesting things. There's a few points here highlighted. First one is that the donors, they really understood or they felt that it's not the same. There's a difference. Donate to hospital use because it was something that was the classical way of thinking. Yes, your blood is needed for patients. But anyway, they were quite happy to do it. But there was some things that we must know. First one, we were very happy to find that they say that yes, we can do it because we trust the blood service system. That is, if they ask for the research, we feel that yes, because they are doing it so well, and we were very proud, of course. Okay, that's good for us and very nice. But then there's also something else. There must be, for example, some kind of transparency or openness. They wanted to know if we collect them for the research. They would like to know what kind of research it is, who is benefiting it, are we getting money out of it, are we selling the sample somewhere, or is it just for the common good? So this was for the biobanking, but it's the same as for the research use, and also there was a lot of issues related to use of the data, health register data as well. And this was something that was very, very good for us, because then, in a way, we would conclude that, yes, our blood donors, at least in Finland, they felt that, yes, that we can donate our samples to research and biobank as well, assuming that there's some kind of transparency. It must be for common good. They were very much against selling it or getting a lot of benefits or a lot of gain or money out of it. And also something that was very good for us to know is that they say that, yes, I want to keep the sample, but perhaps at the same time, if I'm donating the blood anyway, don't ask me to come for the separate research donation at all, because my time is so important. So we concluded, not only based on these, but anyway, we could get some kind of support to say that yes, we can have blood donor biobank because we get some data that yes, the donors accept it. We didn't want to do it if they say that we don't want to have it because we want only to donate blood for the hospital. So we now have it, and something about the biobank system in Finland. We have a special legislation that is regulating quite a lot of the biobanking system. So the biobank in Finland doesn't mean that you have a collection of samples. That in many countries, it means that you have a biobank if you have some samples. It means that you must have some kind of organization. There must be a person who is responsible for the biobanking. There must be quality control. There must be data people and all instructions how you are working. And once you fulfill all the criteria, you get the permit from the authorities to have a biobank. And once you have it, then you can ask, in our case, they are blood donors, but in hospital biobanks, they are patients. You ask them consent for giving the samples. They can be extra samples, or usually they are just the samples that are leftovers. And then also the use of personal data. So it means that you give a biobank consent, giving permit to use your samples and also your personal data if they are in the registries. And then in a biobank, you have samples, basically. And then whoever wants to have samples or data that is inside the biobank can make an application. You can also make an application. The Finnish law is such that anybody can apply, assuming that you have a valid research plan, you have ethical permits and so on. But anybody can do it, and we must keep the samples out if we don't have any reason to refuse. We give samples to the research project. They may combine the results with some registered data. We don't have them here. And the point here is that once, for example, if they get DNA samples from 10,000 blood donors and they are sequencing the whole genome from this, they must return the genome data back to the biobank, and the biobank can give it to the next process, so that in a way we increase the value of the biobank, and there's no need to replicate always the same studies. And also the results must be somehow public. It can be a scientific publication, or it can be some other way, depending a little bit of the process. Of course, if this is a company testing their new drug candidate, they don't need to say what is the drug candidate, but it can be so that they must anyway somehow give out results. Yes, this is the biobank system. And one of the main, sorry, you cannot read. There's a FinnGen project.
You can read that for you while you speak.
Yeah.
Sorry, I just need to get closer.
It was the main, one of the first and the main, don't worry about it.
Oh, sure, yeah. To bring it down a little
There's just title of FinnGen project.
Yeah.
One of the first biobank project we had, and also one of the reasons of to get a permit to start it from the director of the blood service was that there must be somebody who is really needing the samples. And this is a very large project. We have collected 10% of the Finnish population using the biobanks, hospital biobanks, and also blood service biobank, so that there's about 500,000 Finns. We have the genotypes based on the samples collected here at biobanks, and we have the permit to combine the genome data to all health register data we have in practice in Finland. to be able to study the genetics of all diseases we have in Finland. And the point here is that they also wanted to have healthy blood donors as a control, but also for other purposes. And there's 58,000 blood donors who are part of this FinnGen project. It's a biobank project, so they are genotyping it. And then, as said, they must return the results to the biobank. So it means that we have in a blood service at the moment about 50,000 blood donors who are genotyped or who have been genotyped, so that we can use them for other purposes as well. There's a lot of things coming out of this. There's at least something like 500 publications already based on this FinnGen project. And it's one of the largest investment of the biomedical research in Finland. And it's led by Professor Partanen and Mark Dale. Mark Dale is now in Broad Institute. He was then in Finland for 6 years. It's funded by the big pharma also, so they are part of the project. But there's all hospital biobanks also as well.
Control's gone out.
This is the kind of flagship paper we published a year ago in Nature. And the main point is that the Finnish population is a small one. It's a little bit isolated, so that the genetic background of the Finns, it's quite narrow. There's bottlenecks and so on. And the main finding one of the main findings that is of interest to my present talk is here. We could identify a lot of in genome-wide association studies for common diseases. We were able to identify quite a lot of new genetic variants that are associated with these diseases, and they were such that they are enriched in a Finnish population. They are quite rare in the Finnish population as well. Most of them have a frequency of few percent only, but they were enriched in a Finnish population and they were quite strongly associated. For example, just to give you some examples, there's glaucoma here. There's one specific variant. These are the way we are calling its specific location in the genome, and there's a mutation changing arginine at position 220 to sustain in one gene, and this variant is 87 times more frequent in a Finnish population than it is in other populations. And we can see here that is the frequency in Finland is about 4%. Estonia is 1%, and you cannot find it in the UK biobank at all. So it means that, for example, this mutation is such that it can be found in the Finnish population and related Estonian population, but not elsewhere. And so on. If you are interested in immunological diseases, for example, there's IBD and ankylosing spondylitis, there's very highly enriched variant that is in both of these diseases found in Finnish population, but again, not at all in UK biobank. So something that is only in Finland. And what is this, how this is related to the blood banking is sort of the first, it's Jonna Clancy who is doing the thesis. We wanted to see, are these rare variants related to the, can we find them in blood donors at all, because they are quite highly high risk alleles, and we have something called healthy donor effect. We are selecting only healthy people, and we were able to demonstrate that yes, there is the frequency in the blood donors, and this is the frequency in hospital biobank samples, and then the frequencies were very much the same. So we can assume that you can really find these mutations also in healthy blood donors, and in many cases the blood donors have quite many of them, so that about 80% of the blood donors really carry at least one of the variants. So we can do, and what we are doing at the moment, we are resampling, we are taking the new samples from the same donors that have been genotyped in a FinnGen, and we are collecting living blood cells, and we take plasma and serum samples, we freeze them very quickly so that we can do a lot of different kind of omics to be able to study these variants that are enriched in a Finnish population that you cannot find samples from elsewhere. And why in a blood service? The reason is that the regular blood donors are coming to donate every now and then, so you get the samples and we can collect from the whole population from all over Finland. So it's a quite nice way of having samples carrying those mutations, and there's at least 200 such mutations now identified that are enriched in Finnish population, not really found elsewhere. Yes, and the aim is that we can do multi-omics studies and try to identify what is the reason behind these mutations, how they are related to these diseases, and can we find new ways of new targets for drugs and so on. Sorry. Slide, but this is the way what they're doing. We are just not recalling them. We just, when they are coming to donate, we take samples from the drivers and pouch, so that we have living cells. We freeze them in nitrogen, and then we have serum samples for and plasma samples, and also very quickly try to freeze them so that we can get good quality metabolomics and proteomics. And what they're doing at the moment, it's basically Masahiro Kanai, who is working at the Broad Institute in Cambridge, US, in Ramnick's lab, who is doing most of the work. And we are doing multi-omics, so that there's systematics, proteomic metabolomics, then sequencing, immunophenotyping, single cell sequencing, and so on, so that we collect all possible data. And this is just a busy slide made by Masa, who is over there, so that we can see the sequences here, identify the variants, and then having samples from the blood donors who are healthy, so there's no problem related to the drugs or medication, and also the progression of the disease. We have immunophenotypes, we have sequences, we're seeing those sort of things, and one by one we try to understand how they are working, what kind of pathways they are affecting, and so on. Everything is still in progress, so I don't have any real results. So this was the science, how we are utilizing the genome data for science purposes, but then something we are doing inside the blood service. The first one, this is also related to Jonna Clancy's thesis, and she wanted not only to test how the FinnGen enriched mutations are found in blood donors, but also we made a genome-wide association study, so that we took the blood donors and we compared them to the hospital-based samples from the FinnGen. Can we find any genetic markers that are related to the status of blood donation? And And this is basically the result. Here we have all the chromosomes. If you don't know the system, there's a lot of markers here. We have something like 10 million markers imputed from the SNP arrays. And higher the peak is, the more you have evidence that there are. This is one place where these genes are somehow related to blood donation, and that is blood donor status, and also these ones here. There was in total 51 quite high number of signals coming out of this that are related to the being of blood donor. But at least part of the story is very, very simple. The first one is that the highest peaks here are related to the blood groups. So this is ABO, this is RH, and this is KEL, and we are selecting O negatives, for example, for blood donors, and KEL negatives. So it seems to be so that first when you see this kind of Manhattan plot, you are very happy, hey, there's something coming out of it, and then you are checking what it means, one by one you will notice, okay, perhaps it's not so fancy as you were thinking. Anyway, it was interesting. And the second one is related to iron. When you are donating blood, you are also donating quite a lot of your iron. Same time. And then when we went a little bit deeper, the smaller peaks, they are still very strongly associated in a statistical way. There's at least two genetic variants that are related to your iron levels. One is hemochromatosis mutation. It's a disease causing your iron levels are increasing all the time. So most likely, if you have this kind of mutation, you are very good blood donor because your iron levels are held in high levels. And then the other one is related to anemia risk, and it's a mutation that we have. This actually enriched in the Finnish population. I drop out this slide to save some time, but this is one of the Finnish enriched mutations in RNF43 gene. It's 200 times more common in Finland. It's related to iron deficiency anemia. And yes, most likely we are selecting against those persons who carry this mutation because they can be blood donors. And this is also something that's interesting for us. Of the iron, one pilot we are doing at the moment is related to hemochromatosis mutation. And hemochromatosis is a very nice example because in the European populations, most of the patients have the same mutation for some reasons. And it's related to the gene FHE. And we screened from 43,000 blood donors who were homozygous for these specific mutations. We found 94 donors. And then when one gives the biobank concept, we are asking if we are going to find something that is most likely important for your health, can be recontact you. And of these 94, 89 have given the permit that we can recontact them. And then those we selected and we first checked, because this was research based the typing. We didn't know if this result is reliable. We checked the mutation from the same samples. In clinical test, actually found that yes, all of them would be confirmed to carry homozygous mutation. And then we contacted them and we give them new instructions how they are donating blood. We say that yes, you can donate it if you feel, because it's very good for you, because this is if you donate blood, you take also the iron off, and we don't give for them iron supplementation. We in Finland we give in some cases iron supplementation. Interesting story is that only 6 out of these 89 who were homozygous for these mutations knew beforehand that they have this kind of disease. I don't know if it's good or not. It depends a little bit how you think about it. It may be so that, yes, we help them to give the diagnosis, but you can also say that they are totally healthy, they donate blood, and perhaps the penetrance of the mutation is so low that they would never have any problems, but now they know that they may have some problems. So it's a balance always. But this is something that we are now thinking quite a lot. We have a lot of genome data, how much we should get back to blood donors and what kind of implications it is for your life. Because it's not only your data, it's also your family data and your and so on. There's a lot of problems related. In fact, we are just thinking, this is in process. Should we also screen some other mutations like breast cancer BRCA 1 and 2 mutations, and there's a Lynch syndrome that is related to cancer in the intestine, and we are thinking, should we screen these mutations also in blood donors, and what it means if we are doing it, and how we return the data back to the blood donors. Is it good or not? We don't know it at the moment. We have not decided. It's a little bit difficult to know which way to do it. And still more, how we are using the genome data in our, let's say, internal activities, and as the blood types, ABO types, and so they are genetic traits, and also HLA is genetic trait. We can impute, we can predict based on the genome data all the blood types and all the HLA alleles from these, and how to utilize them. First, not all blood types can be directly picked up from the array data. So we set up a machine learning methods how to kind of impute or predict all blood types from the SNP array data. And it's, I don't know if it's published already, but it's accepted. And we did it in our Finnish population, and then we replicate it in the Danish population, so that we demonstrate that basically we get all blood types out from this kind of genome array data. And how to utilize this? We can screen now. We have 58,000 blood donors, of whom we have the genome data, and now we can screen those who have some rare blood types that are needed, perhaps. And this is just some examples. If you, let's say, well, negatives, we knew beforehand that there's 8 blood donors in Finland that are negative for this blood group, and we were able to identify 8 more based on this genome data, so that we can increase the number of blood donors who are having some rare blood types, but not always. For example, this P minus, there's none of them coming out of the data we have at the moment. And say for the HLA, we first set up the imputation method for HLA alleles, and why we want to have HLA alleles is that those of you who are in a blood bank may know that HLA type thrombocytes are needed for leukemic patients, so that they are not immunized. And what we are doing here, we first set up the imputation method, and it has been published already. And then we have in a biobank genotypes. This should be 58,000, and we can impute HLA, or we have imputed HLA alleles from all these individuals, and then the people who are taking care of the HLA type thrombocytes selected, in this case, 2,000 blood donors who were homozygous for some HLA alleles, or they had some HLA types that they were regarding as very useful for HLA thrombocytes. And then they contacted again, are you willing to donate thrombocytes only? Because thrombocyte donation is a little bit different. In Finland, you must live in Helsinki area, and also you must be quite, let's say, big guy or something. I don't know why, but it's that way. And half of them accepted, yes, I can do it because I have the rare HLA type, and that way we could increase the pool of the blood donors instead of typing the HLA alleles directly from all these individuals. It would have been much more expensive. And in fact, if you are selecting HLA homozygotes, you can work with a very low numbers. We were able to demonstrate that there's 48 different HLA AB homozygote haplotypes in Finnish population in this set. And with this, sorry, you cannot see it here.
This is annoying me so much.
But with these samples, we could basically have matching thrombocytes for almost the whole population. It's something like 90% of the population are matched because they are homozygous. So it's and this is also in other populations, but this is perhaps the lowest number I know because the Finnish population is so narrow in a genetic ways. And there's 20,000 people in this study, and there was more than 700 individuals for the homozygous. So if the numbers are quite nice, we could invite all these to donate thrombocytes in a way, so that we could almost always get matched thrombocytes for patients. So try to get this. So to conclude, first, we have evidence that yes, it is accepted by the we ask them to donate for research use and for biobanking. And then once we have in a biobank, at least in Finland, it's so much genome oriented that we have genome data, and it can be used for many research purposes, like in FinnGen, and it helps because the blood donors are willing to donate, and they come donate every year or even many times a year, so you get fresh samples. You can impute or you can predict the blood types of the HLA alleles from the genome data, and this way you can increase the donor pool of rare blood types or HLA thrombocytes. And then there's a question of how to return. Should you return what kind of genome data to these blood donors, and what it means if you are giving back some quite delicate health data back to these donors. So I guess this was my talk, and there's a lot of people who have been working with me, or they have been working independently. Also, they don't care about me so much, but anyway, there's a lot of people who are working with us, and also, if you want to come to Finland, it's a very good opportunity because I used to be the research director of the last service. In the beginning of this month, I was able to drop out of administration staff, and now we are hiring a new staff. So if you want, it's a nice place. It may be a little bit colder than here, but anyway. So thank you.
A very nice overview of what you're doing. Biobank product research. Question from people online or on site. Just maybe I'll start with one. So you mentioned that your basically, once you identify those donors with rare variants, you're then collecting through when they're coming to donate.
Yeah.
You're collecting additional sample for multiomics,
Yes.
single cell analysis and so on. Are you as well collecting serial sample on them, or just
Yeah, that's very good question, because we have been thinking and we are doing it in a small scale at the moment, and it is because we don't have money collect all the time. I mean, you can collect plasma samples, it's quite cheap, but if you want to raise the cells and keep them. So we try, and it's the plan that we would like to have several samples, so that every time they are available, we would have in the end nice set of samples. So if somebody would like to know what happens to your profiles when you are getting older, or if you are getting flu, or if you are doing something like that. But we try to do it, but at the moment in a small scale.
For the single cell study, how many samples? What is the sample size for the single cell studies?
At the moment, we have data from about 1,000, and it may be so that we are in the end having 2,000 samples. It's quite expensive at the moment, but the FinnGen is doing it, like the proteins do, and the results are coming back also. Our biobank in the end.
Thank you very much. And in terms of the donor relationship you have with biobank donors, in terms of who decides what is relevant data to feed back to them, who controls that?
If I would know, nobody knows if we can at this moment what is relevant data to give back. We have been thinking it, and at the moment, we don't have the rules in Finland. And for example, for this returning the hemochromatosis data, we had a lot of discussions with ethical boards, how to do it, and what kind of how they can get advice how to do it in the future with their health and so on. It's quite easy, but if we go to some other disease, it's much more expensive, complicated. Sorry. Yeah. So I don't have.
So those patients with the homozygous allele for chromatosis, they didn't have any. You were testing their hemoglobin at every donation, and it was absent. They were in the normal
They
range.
had just normal. I mean, some of them had relatively high, but there was also something that somebody who were not having very high levels of hemoglobin. So basically, hemochromatosis patients should have high level, but if they were donating all the time, or they were menstruating women, or something. I don't know the details so well, but they didn't have any symptoms. So they were basically recovering themselves as healthy.
Thank you for the so I was curious, obviously, with the case of the hemochromatosis, right, that was donors who I guess are at less or lower risk maybe of iron deficiency and lots of donations. Then that kind of motivates them to call them maybe for more donation and then without medication for iron. But there are cases where also donors would be at high risk of like adverse events, like let's say VR, for example, so fainting. Is there any way to, so obviously you can identify those high risk of that, and then is there any way for the blood service as a whole to try to implement measures to restrict or change donation processes for those donors who are at higher risk of those kind of complications? You know, it's probably logistically challenging, right, to actually implement that.
I don't know if I have any good question for any good answer for your question. How to utilize the genome data is tricky. I mean, I don't know if this is related, but what we have been discussing quite a lot is that the donors, they are coming to donate blood, basically. And then they may give for biobanking, there's E. coli, And then we are screening a lot of things, and just some people in what I would say that we shouldn't much utilize the data because they have come to A plus, and we can do all research and should be identified because basically they are healthy.
Yeah, so I guess the question is whether or how to feedback data. Well, if and so how. That's a big question. Is it something on the chart online?
Yeah, we'll have a look.
Right, in the meantime, so is there any plan to use training data for blood group genotyping? So you show us that you have done some work on that.
Yes, we have.
But is it already put in practice? So is it already used by the blood service?
We are using it, but only for because it's the research tool. So it's not the final proof tool. So we are screening. What they're doing just at the moment, we are screening 60,000 blood types, and we are trying to identify all those that are of interest to blood service. And then when they are coming next, we are typing using the accredited clinical protein test, because otherwise we cannot use it for clinical purposes. It's the same as in the HLA finding. It's only for the research use because it's not accepted to be for real clinical.
We have a question online. I have to repeat it because the sound won't come in. Thank you, Roxanne. Roxanne Sterling is one of our patient involvement panel members, and she was asking, does the Finnish Blood Service routinely give iron supplements?
Yes and no. We give it those who have low hemoglobin, and also we give to young women because we know that young women have very low, they have high frequency of low hemoglobin, and also we have measured some ferritin levels, so the iron levels are quite low. So we have decided that we give the supplementation, but we just give the pills and we don't know if we are taking them or something else, but that's what we are doing. I know that not in all countries.
Not here.
Not yet, at least. And we are feeling that it's working quite nicely, but we don't know exactly because we were talking already that we have not tested everything, so we don't know. We changed a little bit the policy for young women, but we don't have data how well it's working in real life.
Great.
Still,
Is the linkage to the health records something already established, or is it established on a project basis?
It depends what you mean, but if you want, let's say, if you would like to have a biobank project in Finland and you apply samples from us, then you must get the registry data from the registries. So you must make an application to the registries. In a FinnGen project, there everything is working out. So we don't have any access to health registries.
so every time need to be so once you have
Yes,
a project,
it's a project that is having the data. We can only provide what is inside the blood bank. In a biobank, meaning samples, there's genome data and something that is directly measured from the sample. That's the law saying that we can have only the data that is from the sample, not from, let's say, from the cancer registers. But you can, of course, try to get it from the cancer register for your project. I don't know if this
So you apply to the main registry and then you say, I need it linked to blood donors.
Yeah.
Let's say if you want blood donors, or if you wanted the general population.
Yes, you must have permits so that you can link this. And as we all know, it's turning more and more difficult to get all these things working so that you can really combine all the data. But the biobank law is quite practical here. If you have a biobank process, you have more rights to combine everything than if you have just the standard resource. That's the Finnish.
Smart question.
Sorry to ask you again. Maybe I missed it, but what are some of the basic demographics of the biobank donors? I mean,
You didn't miss it because I didn't say it. But the biobank donors are a little bit older than standard blood donors, and they are more regular. So it seems to be so that those who are really committed to be blood donors also want to keep be a biobank donor as well.
So is that an area that you want to sort of, kind of work on? Or?
I would like to do it, but our donation people say that they don't want the first time donors because there's a lot of things for them, telling what to do, and they don't want to ask, do you want to sort of biobank? So the youngest one, they are a little bit, they are missing it.
Excellent. Well, thanks again, Jukka, and thanks for the.
Thank you.